please help me welcome tonight's lecturer, Anita Metzler.
Thank you, Gina. Thank you, everyone, for spending your Thursday night listening to me talk about lobsters. I'm going to apologize. My voice is a little raspy. I lost my voice over the weekend, and it's taken a while for it to come back, so I apologize if I sound kind of bizarre right now. So as Gina mentioned, I am an assistant scientist and laboratory operations manager, which is a mouthful, in our research department here at the aquarium. It's part of the newly minted John H. Prescott Marine Laboratory, which, if you're not familiar, we actually have a laboratory on the 5th floor of the main building of the aquarium. It's completely off exhibit. It's not in public eye, but there is actually a 5th floor to the aquarium. So I'm going to be talking about this guy, the American lobster, and his cousin, the European lobster, tonight. So the title of this talk was Lobsters from Petri Dish to Dinner Plate, which is a really exciting title, but unfortunately that is actually not what I'll be talking about, mainly because no one actually does this. So no one grows lobsters from a Petri dish and grows them to adult size for us to eat. So we don't do that here, and it's not something that anyone else does. So if this is what you're hoping for, I'm really sorry to disappoint you, but that's not what we'll be talking about today. So what we will be talking about is I'll do a little run through about the aquarium's lobster research facility, which I manage. And I'm going to talk a lot about our research, which focuses on shell disease. I'll touch a little bit about our past temperature work that we've done, and then I'm going to continue on talking about some work we've done with European lobsters. So if you've never seen our lobster research facility, we run 2 very small rooms. This is one of them. This is the bigger of the 2 rooms. It's quite small still. We raise baby lobsters from eggs. We basically get egg-bearing females from the Massachusetts Department of Marine Resources or from fellow colleagues, some of who are in this room. This is what the eggs look like. This one's about ready to hatch. It has quite a large eye forming right here. It also has a very small yolk sac. That's how I know it's about to hatch. When they do hatch, they go through a larval stage. They hatch looking like this little guy. These are about the size about the end of a pencil. They're quite small. They go through 3 larval stages, and this is what they look like live. They tend to be kind of bluish. After that 3rd larval stage, they go through a metamorphosis, and basically that's when they start looking like a little tiny lobster. In the wild, the larval stages are planktonic. They're kind of at the ocean's whim. They're just kind of floating around the ocean. But the post-larval stage, this what's called stage 4, is when they start actively looking for a place to live on the bottom. So they start swimming down to the bottom and looking for a place to live. And once they find a place to live on the bottom that they like, they're going to be benefit for the rest of their life. They live on the bottom the rest of their life. In the laboratory, when they reach this stage, this is when we take them out of sort of a communal larval rearing tank, and we start holding them individually. And that's pretty much what it looks like. We hold them in small containers. These are actually medicine cups that you would take your cough syrup in, with a whole bunch of holes drilled in them. Some poor intern had to do that at some here. We hold them individually. They get an identification number. And this way we can track how often they're molting, how often they're shedding their shell, which ones are related to each other, and just kind of monitor their progress. As they get older, we just kind of move them into bigger and bigger containers. Now, it's important to keep them separate by individual, because otherwise this is what happens. They like to fight each other. American lobsters are they're incredibly antagonistic, and they're incredibly cannibalistic. And even at the larval stages, they're cannibalizing each other. So if you want to actually keep any of your animals, you actually do want to separate them as best as possible. Otherwise, they'll do this, maybe without the butternuts, but something like this. So lobsters can be born a whole bunch of different colors. And this is just a variety of some of the colors they can be born as. And this is a genetic mutation. But a normal colored lobster, their shell color is all based on one pigment that they have to eat. So when you think of your normal lobster sitting in a lobster tank, it's sort of a brownish color. And that coloration is just all based on this pigment called astaxanthin that they have to eat. The way astaxanthin works is it's all by itself, it's red. It's in its free form. And as a lobster starts eating the astaxanthin, it starts incorporating it into different parts of its body. So when it starts eating the astaxanthin, it'll first incorporate it into its muscle layer or its tissue layer. And in the tissue layer, it stays in its free form and it stays red. If you keep feeding the lobster astaxanthin, it starts building it up into its body, and it'll start moving it into different parts of its shell. So it'll incorporate it up into the shell, the first layer being the layer closest to that muscle layer. And once it moves into the shell, it has to bind to a protein. And when that happens, the protein astaxanthin binding causes the astaxanthin to kind of twist a little bit, And instead of being red, it shifts and turns blue. So now we have a red layer and a blue layer in the lobster. If you keep feeding the lobster astaxanthin, they'll incorporate it even more, and they'll move it into the top layer of the shell. And when that happens, it binds to a different protein. It shifts a different way, and it turns from red to yellow. So basically what happens is a normal colored lobster has a red, blue, and a yellow layer, and our eye can't handle all those different layers, and it basically just kind of mushes it all together, and you see brown. This is also what I tell everyone is your dinner party tidbit. So the next time you're schmoozing with people and you want to impress them with some like fascinating knowledge about lobsters, because who doesn't want to do that? This is the why lobsters turn red when you cook them. So when you cook a lobster, the heat from the water basically causes those proteins to denature. They fall apart. And when they fall apart, they let go of that astaxanthin, they let go of the pigment, and the pigment goes back to its original form, which is red. So that's why they cook red. So next time, you can wow everybody. So the reason why I'm telling you this is in a laboratory, we sort of stumbled upon this. This was actually known, lobster researchers discovered this a couple decades ago. But in our laboratory, we sort of stumbled upon this by accident. And what we found out is if you feed a lobster a diet that has very little astaxanthin in it, you actually get a white lobster, which was really cool. We found out if you only give them a little bit of astaxanthin, they'll turn blue, and then if you give them a whole bunch, you get a normal colored lobster. We stumbled upon this completely by accident. Basically, we found a food that was used for feeding to shrimp, and it was nice and cheap. And so we're like, oh, great, shrimp, lobsters, let's feed it to them. And after a while, they were like, our lobsters are a little pale. What's going on here? And we found out that the reason why the food is cheap is because they don't add this pigment to it. So I'm telling you all of this not because this is going to be a focus of our talk, but you're going to see a lot of lobster pictures, and a lot of them are going to be sort of the blue or white lobsters that you'll see. So just to sort of allay those questions, you know why they're blue and white. So when it comes to research, I'm going to talk mostly about epizootic shell disease. And again, I'm going to talk a little bit about our past work with temperature, and then I'm going to move on to our more current work with looking at European lobsters. So epizootic shell disease. So this picture is of a normal, healthy lobster. Everyone kind of knows what that looks like if you're from around here. This is a lobster with shell disease. And if any of you kind of looked at that and went, oh, gross, that's exactly the reaction that we are looking for. Epizootic shell disease is primarily a bacteria that settles on the lobster's shell and literally just starts eating away at that shell. So in this picture, what you're seeing is all these little pits that are formed. This is all bacteria that have eaten away at the lobster shell. The sort of brown-gray areas, that's actually the healthy part of the shell. So this is a pretty severe case on an adult lobster. The interesting thing about shell disease as a whole is that it's actually been around. They first identified shell disease as a problem or as a disease of lobsters in the 1930s. So it's not a new disease, the concept of shell disease in general. It's not new. There's a lot of research that was done in the 70s on it, but in about the past, I would say about 15 to 20 years, we're starting to see a sort of an outbreak of this form of shell disease. And it started off in Rhode Island and Long Island Sound and has slowly spread. It can be problematic for the lobster population. It can cause mortality, not always, and actually not usually, but it can cause death. The problem is with the lobster fishery. If you're a lobsterman and you want to sell a lobster, if you catch this guy, you're not really going to sell that for a premium price. If you're an average consumer, if you have a choice between this lobster and this lobster, good chances are you're not going to pick the disease-looking one. You can actually eat this lobster. There's actually no problem eating these guys, and there have been lobster biologists who have tested this theory. They cook up a healthy-looking lobster and then a diseased lobster, and they say, oh, it tastes fine. So I haven't done myself, but I would have no problem eating a lobster with shell disease. But for the most part, average consumer, you're not going to spend your however much money on a lobster that looks like this. So it can affect the lobster fishery, and as I mentioned, it can affect the wild population to a certain degree. In a laboratory, this is what shell disease normally looks like. This is one of our juvenile lobsters that were raised here at the aquarium. If you look really closely, you can see some darker spots forming in between its eyes, what's called its rostrum, and then also right behind its eyes, the claws are also being affected here. What's really interesting is in our laboratory, since we work with these color change lobsters, is you can see the disease that much easier. So it became a really unique laboratory model that we worked with these white lobsters and blue lobsters that we can start identifying shell disease at very early stages. So for example, if you take a white lobster that we've created by diet, and you start noticing shell disease forming on it. Now all these brown spots that you're seeing, this is the start of shell disease. And what I like to point out is, first of all, this is very early stages. The brown spots are noticeable, but if you were to look closely at this lobster, you wouldn't see any shell erosion happening just yet. The brown spots themselves are not actually from the disease. It's not the bacteria that's causing that brown. It's actually the lobster's immune response to something attacking its shell. So basically, the lobster knows something's there, and it's doing what's called a melanization response. It's trying to actively prevent something bad from happening. So this same lobster, 2 weeks later, you can monitor it, and you can see how the disease is getting worse. So that rostrum area, that area between the eyes, that's now formed into a lesion. You can see some visible erosion. Same thing with some of the claw areas. Those are starting to be worn away, along with the tail. You'll notice that this area on the carapace, on the body, it's actually being, it's getting worse, but it's not. It doesn't seem to be forming lesions, but you can see there's just more spawning happening. This same lobster, 2 weeks later, gets even worse. It actually gets so stressed out that it decides to eject one of its claws. Don't worry, he can grow it back. And you can see the lesions have gotten significantly worse. 2 weeks after this, or within a 2-week period, this lobster molts. And when it molts, it sheds the shell that's all diseased and gross looking, and gets rid of it and forms a whole new shell. And the shell is clean and free of disease at the moment. So the interesting thing about shell disease is that it gets progressively worse over time. It's usually worse right before it molts. And that after a lobster molts, they'll have a clean shell for at least a certain amount of time, but they're in the environment that has that bacteria that's settling on the shell and creating more shell disease. So over time, this lobster will get shell disease again, or can get shell disease again. So we study shell disease in our lab, which some people find really interesting. They're like, well, how do you study shell disease in your laboratory? So we run a recirculating system. We basically bring water in from the aquarium, and then, from the harbor, and we recirculate it around and around, every now and then always bringing in some new water just to promote some water exchange. In this process, we have built up a high level of bacteria. So our lobsters just in the lab just get shell disease on their own without us doing anything. They just will form it. Now we always work with juveniles. They're relatively young, only a couple years old at most, and they molt about every couple months. They're molting very frequently for a lobster. An adult lobster usually molts only about once a year at best. So because our lobsters molt so frequently, they will get shell disease, then they molt, and then they're okay again. So once we started getting interested in looking at shell disease, we decided, well, hey, let's start studying this disease. You can sterilize seawater in a system like this. You can try to kill the bacteria that's in your system. But once we decided that we wanted to start studying shell disease, we said, well, we want to study the bacteria, so let's not kill the bacteria. I would like to point out that our lobsters never get like this adult lobster. They never end up looking this bad, most likely because they are molting way too often for it to get this bad. So this is pretty much as bad as it gets with our lobsters. So what exactly causes shell disease? And this is a really big question in the lobster biology world. And the answer is no one really knows for sure, but the general consensus is that it's multiple factors. The first is that you need to have something causing the disease. In this case, it's believed to be bacteria, but no one knows for sure exactly what bacteria it is, if it's one bacteria or if it's a suite of bacteria that needs to be there. There's also probably an environmental stressor that needs to occur, whether that be a pH change, pollutants in the water, temperature changes, any number of things that can affect the environment that the lobster is in. And then there's the lobster themselves. Maybe they're immune compromised. Maybe they're not in the best shape right now. Maybe they just got attacked by another lobster and they're not feeling so hot. So any number of things can be going on with the lobster. And once you have sort of this suite of things happening, that's when you'll see disease. The other interesting thing about shell disease is that it's not exactly contagious. We always like to relate it to the common cold. So just because one lobster has shell disease does not mean that the lobster next to it is going to have shell disease. It can, but it's not necessarily going to contract it from the other lobster. So if I have a cold, one of you might get my cold, but most of you probably won't. So again, it sort of depends on maybe you're stressed out from work, then maybe you're more likely to get my cold. But if you're in perfectly good health, you may not get my cold. So that's sort of the way we relate how shell disease is. It's contagious to a certain degree, but not in like widespread contagion. So our laboratory, when it comes to trying to identify what causes shell disease, we've looked at a couple of different things. We've looked at diet effects, we looked at temperature, and we looked at the causative agent, or tried to identify the causative agent. With diet effects, we basically found out that diet can affect lobsters, and it can affect their health and affect the amount of shell disease they got. And I'm not going to really talk about this today, but I just want to give you a little bit of background that we have done work on this. When it comes to identifying the causative agent, we have a couple of theories about it, but that's still really up in the air in the lobster world. And again, I'm not going to touch too much on this, but I'll talk a little bit more about identifying the pathogen a little later. What I do want to kind of brush you up on is the work that we've done on temperature, just to give you some background as to the work we've done. We are interested in temperature because shell disease, epizootic shell disease, first showed up in Rhode Island and Long Island Sound, and this was in the mid-90s. And since then it kind of spread northwards, north of Cape Cod into Boston Harbor. You can see it in Cape Ann and the Gloucester area. But once you start getting into New Hampshire and Maine, you pretty much don't see it. So there seems to be a temperature issue. If you're from around here, you kind of know that, that south of Cape Cod, the water is much warmer because of the Gulf Stream, the influence of the Gulf Stream. Once you get north of Cape Cod, the water gets significantly colder. And then once you kind of get into the New Hampshire, Maine area, it gets even colder. So again, you kind of know in the Maine area, the water never gets exactly warm, or what you would call warm, or what I would call warm. So we were really interested in this, this temperature issue and whether temperature would had an effect on the amount of shell disease lobsters we get. So the theory that or the hypothesis that we had was with increasing temperature, you would have an increase in disease level, right? So the warmer the water, the more active the bacteria. The bacteria are going to eat away at the lobster shell and create more disease. The colder the temperature, the bacteria are going to be slower, they're not going to be as active, and you'll see less disease. That was the hypothesis we ran at. So we had lobsters in our lab. We kept them at 3 different temperatures. We kept them at 10, 15, and 20 degrees Celsius, which works out to be about 50, 60, and 68 degrees Fahrenheit. And we held them like this for quite a long time. And the average amount of shell disease over time worked out to be a little bit different than what we thought. So this is just basically a disease index. And at the 3 temperatures, what you're seeing here is at 15 degrees is where we're seeing the highest amount of shell disease, which kind of surprised us. We really thought it was going to be at that 20 degree, that warmest temperature, that you would see the most disease. So what we kind of forgot about is that we do look at juveniles. And as I mentioned, juveniles tend to molt pretty frequently. So if you looked at the average molt cycle, basically the number of days it takes for a lobster to go from one molt to the next, lobsters are highly influenced by their environment, and temperature will increase their molt cycle, which was something we probably should have thought about before we started this experiment, but didn't realize until after the fact. And what you'll notice is that at 20 degrees, they have the shortest molt cycle. They basically take the shortest amount of time to go from one molt to the next. And at 10 degrees, they have the longest molt cycle. So basically what we saw was a confounding factor. And basically the lobsters at 20 degrees were getting shell disease, but they were molting frequently enough that they were getting out of their old shell before the disease could get really, really bad. The lobsters at 10 degrees took a really long time to molt, but because the bacteria are a little bit slower at 10 degrees, they weren't getting very bad shell disease. And at 15 degrees, you basically saw sort of a nice combination of the two. The lobsters were not molting too quickly. They were you had active bacteria at that temperature, and so they were seeing a slightly increase in disease. If we correct for the molt cycle, when we look at the disease levels, this is what we get. Basically, the 15 and 20 start looking about the same, and at 10 degrees, we still see a lower amount of shell disease. So I like to call this the Goldilocks effect. Basically, you need the right combination of factors. You need bacteria that are active. You need the lobsters to keep their shell long enough for the bacteria to do its thing, and that's whenever you're going to see a lot of shell disease. Okay, so that's the temperature aspect. European lobsters, what's the deal with these guys? So let me grab a drink of water real quick. Excuse me. So we got interested in European lobsters because we started collaborating with a group of researchers over at Swansea University. Swansea is over in Wales. I had to look this up. I had no idea where Swansea was. It's in Wales, which is in the UK. This is a picture of the university they sent me, which looks like a lovely university. And they had an interest in looking at shell disease with European lobsters. So a little background on European lobsters. European lobsters are pretty much the only other species of clawed lobsters. When you think of the number of species of lobsters in the world, most of them don't actually have claws. They're the sort of the spiny lobsters that you'll see in the tropic waters. The only other lobster that looks like our lobster, the American lobster, is the European lobster. They look pretty identical. European lobsters tend to be slightly darker colored. And from what I understand, morphologically, if you want to identify the difference between the two, it's actually really difficult to do. I believe the European lobsters, I think, have an extra spine on their rostrum, but that's not the best way to identify them. That can be pretty not always accurate. And if some of you are wondering, this American lobster has its crusher claw on the right side, whereas this European lobster has the crusher claw on the left, that bigger claw. Lobster handedness, the claw that becomes the crusher claw, is completely arbitrary. So it can be on the right side or it can be on the left side, it doesn't matter. It's sort of 50-50, so that's also not a way of identifying them. So usually if you want to identify the difference between the two, you have to do it by genetics. So they're, again, pretty much identical. European lobsters, this is their distribution over in Europe, anywhere from Norway all the way down into the Mediterranean Sea. Once you get into the inner Mediterranean, they're pretty rare. And the European lobster in general is a fairly decimated population. They were completely overfished. It was an unregulated fishery. They didn't have the sort of the regulations that we have here for the American lobsters, so they pretty much depleted their population. So there's actually a lot of interest over there to try to restock the wild population. So they actually have a couple of hatcheries where they raise baby lobsters similar to the way we do here, raise them to a certain juvenile stage and then release them out into the wild. So there's a lot of concern about the wild population there just because it's in such low numbers. And so that's where the interest came in with shell disease. Basically, American lobsters are often sent over to Europe for the very same reason that we like to have American lobsters, is because they want to eat them. And the problem is that an American lobster, even if it looks healthy, can still carry bacteria on its shell. And so they're worried about shell disease getting incorporated into European waters. And the reason why this is a concern, because you think, well, if they're just sending them over there to eat them, why would that ever get into the water? Is they actually have found an American lobster. Every now and then they find them, certain individuals in European waters. They do actually have records of this. And most likely they believe this happens by accident in the import process. They also think it's most likely if you have a well-meaning citizen who sees a lobster in a tank and they decide to save the lobster, and they'll take the lobster, buy it, and release it into European waters, not knowing that they're basically just introducing a species that doesn't belong in European waters. Luckily, they never sell shell disease lobsters over to Europe. There would be no profitable economic reason to sell a diseased lobster over to Europe. It's a pretty rough trip. They want to kind of send their best over there. But just because a lobster looks healthy does not mean that it doesn't have bacteria. It's also possible that it's just asymptomatic, that it has bacteria and just hasn't started showing that disease level yet. So the concern is that we have these American lobsters that we're sending over there. They might have bacteria on them. They might be getting released into European waters. So do European lobsters, can they get shell disease? Right now they're not seeing shell disease in European lobsters, but again, the concern is, can they get it? So the way we decided to tackle this question is, with our collaborators over at Swansea University, is they actually, as I mentioned, they hatchery, there's a couple of European lobster hatcheries over there, and there's a hatchery not far from their university in Padstow, England, that raises European lobsters. And they said, okay, well, let's get some of these lobsters that are hatchery raised, they're juveniles, and we'll keep some of them here at our university, and we're going to send you some. And let's just see if they get shell disease. Because we know our lobsters here in our lab will get shell disease. So if we, someone really wants to be here. So if we send you some of our European lobsters, let's find out if they'll also get shell disease. And I said, great. In theory, the ideal way of doing this experiment would be to send some of our lobsters over to them, but we decided that was probably not a great idea since our lobsters are known to have shell disease. It seemed like a good idea not to introduce something that they might not want over in Europe. So, so we decided to only do this one way. And so this is what it looks like. This is our lobster, one of our lobsters that we raised here. This is a European lobster that they sent us. Again, they're juveniles. They tended to be a little bit bigger than our lobsters, so a little bit older. They were kept in slightly different containers. The coloration, I'd like to point out, the blue color that the European lobsters are, I believe this is strictly by diet. I'm pretty sure European lobsters are not naturally this color. But they were really pretty. I was super excited. This is the way the tank looks. So the European lobsters over here, the American lobsters are down there. And so we get these lobsters, and this was about mid-August, mid to end of August. And we received them, we decided, okay, let's just get them into the tank, and we're gonna wait a couple weeks. And then we'll observe them and see how they're doing. So after about 2 weeks, I start looking at the animals and just trying to get an idea of how healthy they are, like what they look like, get some measurements on them. And immediately, I saw this. Look at that, they have shell disease. What do you know? And we're pretty sure that in the 2 weeks that we were holding them, they didn't all of a sudden just develop all of this disease. So, so I called our collaborators over in the UK, and I was like, so they already have shell disease, the lobsters that you sent us. So, and they were like, and I sent them some pictures. I was like, you know, this stuff, and they're like, oh, those black spots? And I was like, yeah, the black spots. And they're like, oh yeah, no, we knew that they get those. And I was like, well, when you were wondering if they get shell disease, that's pretty much what we're talking about, isn't it? So what they were really hoping for is they really wanted to see this. They were hoping to see this on the lobsters. This is what they were aiming for. And I kind of had to tell them that, well, in a laboratory, especially with juveniles, this is pretty much all we get. So the question as to whether European lobsters get shell disease was answered really, really quickly. And so then we're like, okay, well, we have this grant, and we're supposed to have these animals for a few months. What are we going to do for the next couple months? So we sort of brainstormed and came up with a whole suite of questions that we decided to start asking instead. So this was what happens when scientists get together, by the way. I would call it a communication breakdown, except they're Welsh, so it's not like they speak a different language. So these are the questions that we decided to try to come up with, and this is actually expanded a little bit more, but this is all I'll be talking about today. So the first question we decided to ask is, okay, we know European lobsters can have shell disease. So, but how bad does it get? Are they more resistant to it? Do they get it? Are they more susceptible to it? You know, in comparison to the American lobsters, how much worse or how much better do they get shell disease? The second question we decided to go with is, how does the bacterial community in the shell disease lesions, how does that change over time? And is it possible for us to identify the causative agent? This last question was really, that's kind of a shot in the dark. We're really aiming high with that one, but we decided if we can try to answer that question, we should try to answer it. So, so let's start with this next question. How badly do European lobsters get shell disease? And the way to do this is pretty much what we've done here in our laboratory. So we're pretty used to doing this, is we basically monitor the lobsters every 2 weeks, and we keep track of their disease levels. And this is what this looks like. This is basically just me staring at lobsters intently for hours at a time. So if you ever come and visit our lab, this is usually what you see me doing. And yes, I get paid to do this, by the way. This is my job. I had a coworker who brought up the fact that she was watching me. She walked by and she watched me holding this lobster and like looking at its shell disease and said, you know, in any other workplace, this would seem really weird, but at the aquarium, this is pretty normal. We take a lot of pictures, as you know, as you can see all the lobster pictures that I'm showing you. I take an awful lot of photos. How do you call them mine? It's like, I don't know, it's very sporadic. So you monitor the lobsters every 2 weeks and mark where their shell disease is. So the way this works, we've been looking at shell disease for a while, and we've tried a couple different techniques when it comes to trying to track the disease levels. And when we first started doing this, we wanted to look at number. So we're like, okay, you have 2 lesions, great. So let's just count how many lesions or spots that the lobster has. We figured out really quickly that this was a horrible way of studying shell disease, mainly because over time, these lesions can get bigger, especially if they're close by each other, and basically form one giant lesion. So you've gone from 2 lesions down to 1. and which makes it seem like, if you're just looking at numbers, that makes it look like the lobster's getting better, but they're actually getting worse. We've also done percent coverage, where we try to basically get an idea of how much of the lobster's body was covered in shell disease. When we tried to do that, we found that to be incredibly painstakingly time-consuming, to the point that it's taken years actually to analyze some of that data. So we nixed that, and we moved on to number of body areas. So basically, we break the lobster up into a whole bunch of different body parts, and then count how many of those body parts have shell disease. So in this case, this lobster has 1, 2, 3, 4, 5, 6 areas with disease. And if I was looking at only the top part of the lobster, I would actually break this lobster up into about 40 lobster areas. So that's kind of a lot. When we did this for this experiment, we actually looked at both the top side, the dorsal side of the animal. We also looked at the ventral side, or the underside of the animal, and also all the legs. The great thing about working with a decapod crustacean, an animal that has 10 legs that is segmented, is it's a lot of lobster areas. So it broke down to 123 body areas, which is not a lot. The number of areas, so we would count how many areas had shell disease, and then we would calculate a percentage of areas affected. The reason why we did a percentage is because you might have a lobster that was missing a leg or missing a claw, which would immediately subtract the number of body areas that could get disease. And then we also only looked at the maximum disease level. So basically, in the course of the experiment, we found the highest level of disease that lobster had. And then we compared that across all of the lobsters, and then we compared it by species and by location. And again, usually maximum disease level happens right before they bolt. So, and just to remind you, we were doing this here with the American lobsters that we raised here. We did this with the European lobsters that were sent to us. And then over at Swansea University, they were doing this with lobsters that they had kept in their laboratory. And just so you know, we actually had a postdoctoral researcher from Swansea come visit us so that she could learn how we monitor shell disease and make sure that she knew what we considered to be disease and that we were all on the same page on how we were doing this. So basically that we are consistent in our methodology. So this is the way this worked out. What you're seeing here is the percentage of lobster areas that have disease. The European lobsters that were held in Swansea, the European lobsters that were held here at the England Aquarium, and the American lobsters that were held here at the aquarium. And basically the main thing I want you to take from this is that the European lobsters in both locations has significantly more disease than the American lobsters that we have here. So this was interesting. It was actually really nice to see this data work out this way because this is what I thought was happening just from looking at the animals. The European lobsters seem to get more disease, so it's nice to see that that came out in the numbers. And also, just qualitatively, when I was handling these animals, the European lobsters seem to have a thinner shell, and also their shell was, for lack of a better word, they were bumpier. They had more of a texture to them. And bacteria really like a interesting topography. They like sort of crevices and nooks. So it seemed like a thing that a bacteria would really like to settle on. So, so this was kind of nice to see. If you look at all of the lobster body areas, all 123 of them, the numbers kind of work out a little bit differently, but it's still the same concept that the European lobsters themselves, either in both locations, had significantly more disease than the American lobsters. So we found out that European lobsters do get shell disease. They can get it even worse than the lobsters, than the American lobsters. So, so that is not good news if shell disease ever starts showing up in the European lobsters in the wild. But this is all in a laboratory, so hopefully it stays that way. Now, identifying the bacteria community in the shell over time. This was a trickier process. What we wanted to do is, or what, especially what our collaborators wanted to do, was try to identify bacteria on the lobster shell, not only once they start forming shell disease lesions, but at the very beginning. So once the disease starts forming from that initial stage, and then as it progresses and gets worse over time. The problem about this is you never know where and when shell disease is going to happen. It can happen on a claw, it can happen 4 weeks from now, it can happen tomorrow. So how are we going to get that initial stage? We had done work in our lab where we were successfully able to induce shell disease. And we've done this in the past where we basically take some sandpaper and you just scratch at the lobster shell. And if you wait about a week, you'll start seeing disease start to form where we scratch the shell. So basically our answer to how we were going to do this was violence. We were going to attack the animal with sandpaper and tools. And so basically that's exactly what we did. We abraded one side of the carapace with sandpaper. We also punctured one claw with this is going to sound really horrible, but with a screwdriver. And we basically just routinely took samples in order to monitor the bacterial community over time. And this is what this looks like. And once again, this is my job. So what I'm going to show you next is a progression of pictures. This is all of the same lobster. This is a European lobster. To orient you, so on the left side would be its head. And what you're seeing is its carapace, where we abraded it. And on the right side is what you think of as the tail, or it's technically the abdomen. This spot that you're seeing right there, that's actually a lesion that started forming on its own. This is not due to the abrasion that it caused. But what you'll see in this series of pictures is that lesion will get worse over time. So we abraded the lobster's carapace, and about a week later, we checked on it, made sure, okay, great, we're actually seeing a response. We did exactly what we wanted to do. And the next set of pictures is going to be the same lobster at 2-week intervals. And you'll watch as the abraded area gets worse and worse over time. You'll also see that that naturally formed lesion also gets worse over time. So we were successful in doing exactly what we wanted to do. We caused shell disease, and we're able to monitor its progress as it gets worse. And in the meantime, we were also collecting samples for bacteria analysis. The puncture worked out somewhat similarly. This is that same lobster, it's its right claw. We punctured it. Still not working. So we punctured this right claw. 1 week later, we got a response. And the only thing that's a little bit different about this time series is what you'll notice is that this puncture wound doesn't actually get any worse. It changes color a little bit. But the wound, we sort of thought maybe the wound would actually get deeper, that you might start seeing some sort of progression of the wound going into the shell as maybe bacteria starts eating away where that wound is. But it actually pretty much stayed the same. The lobster sort of formed like a little membrane to kind of protect itself, and it just kind of stayed in that same state. So we never saw that get any worse over time. So when it came to sampling the lobsters, we wanted to get bacteria samples every couple of weeks. The question was, is how do you collect samples from these animals in these damaged areas? And our collaborators said, well, they had success getting samples using sandpaper. And I was like, well, that's great, except we use sandpaper to damage the shell. We don't really want to damage the shell any further. So how are we going to do this? We want to get samples without creating further damage to the animal. So the next answer was sterile cotton swabs, which sounds really great, except they weren't really sure it was going to work. So in order to sort of rectify this kind of concept of, well, we need to collect samples, but we also want to make sure this works and we can get DNA from these samples, is with the abraded area, we only swab the front half of the area. The back half of it, we actually use we use a sterile sandpaper. Yay! Okay. So the back half, we always use some sterile sandpaper. The front half, we use a cotton swab. With the puncture wound on the claw, we always use just a swab. It was basically too difficult to try to get sandpaper in there. And again, we didn't want to create further damage to that puncture wound. So we only used a cotton swab. So every lobster that we had here, the American lobsters and the European lobsters, and then the European lobsters over at Swansea, they were all punctured, they were all abraded. We collected samples initially. We also got what we considered called healthy or asymptomatic samples from that same animal, both on its carapace and on its claw. And we basically, every lobster was monitored every couple of weeks. We then took our group of lobsters and we divided them in half. Half of them were sampled every 2 weeks, and the other half we just kind of checked on them, but we didn't do anything until the very end of the experiment. Basically, because we weren't really sure of the swabbing and sandpapering, how much that was actually removing bacteria from our samples, from the lobster, and because we're interested in this bacterial community, we didn't want to interfere with the progression of shell disease. So we actually also just left some lobsters be and just waited until the end of our experiment, took some final samples, and we'll compare this final sample with these lobsters' final samples and see if they kind of match up. So what happened with these samples? I took our samples and I sent them over to Swansea, and they're going to do a DNA analysis. Basically, they take the samples, they extract all the microbial DNA that they can. They amplify a certain area of ribosomal DNA, bacterial ribosomal DNA. They amplify it, so you have lots of them. And then they look at it on a temperature gel electrophoresis, which sounds really impressive, and I'm going to pretend like I kind of know what I'm talking about when I show you the gel electrophoresis. So basically, the way this works is every column that you see here represents one sample from a lobster. Every dark band that you see represents a different set of DNA, and basically the way, in theory, is each band should be a different microbial, different bacteria species. So the interesting thing to note here is the lanes that are labeled with an H, so H1, 2, 3, and 4, these are all healthy shell or asymptomatic shell samples. And the blue boxes that you see here, these are all bands that were only found in the healthy shell. As soon as you start looking at lesions, you don't see these bands anymore, which indicate that these bacteria are only located on healthy shell. As soon as the lobster starts forming a lesion, these bacteria go away. So these bacteria might be beneficial to the lobster. They might be important to the lobster's natural biofilm. We don't really know. And so kind of the next step to this is they're going to take these bacteria and they're going to get the DNA sequenced so we can identify which specific species these are. When they look at the column that says L, this is a lesioned area. This is from a lobster that was, whose carapace was abraded. That abrasion turned into a lesion over time. And the yellow box represents a couple of lobster bands, a couple of bacteria bands that are only seen in this lesion. As you can see in the healthy shell, you don't see those bands anymore, anywhere. So these bacteria are basically only showing up in the lesions area. They're not on the healthy shell. They might be important in causing gill disease. They might be opportunistic. They might just show up because there's a nice place for bacteria to live in and they want to be there. So again, they're going to sequence this DNA and find out which bacteria these are and try to figure out if these are important players in gill disease. The lane labeled P is for a punctured claw, and they basically didn't see anything of interest. To me, this just looks like a whole bunch of pretty bands. I don't know if they're of interest, but according to them, there's nothing of interest. This sequence over here, this A1, 2, 3, 4, and 5, this is all from the same lobster over time. So I believe A1 and 2 is right before and after the lobster was abraded. A3 happened, was a sample taken 2 weeks later, A4 is 4 weeks later, and A5 is 6 weeks later. And the thing of interest is that at A5, you start seeing basically the same sequence of bands that was seen in this lesions carapace show up at that 6-week period. So again, we're not sure if this is a causative agent. It could be, or it could just simply be something opportunistic. But so time will tell on this one. So again, that's pretty much where we're at this point with this experiment. We're still waiting on the final DNA analysis from our colleagues over at Swansea. When it comes to this last question of can we identify the causative agent, I don't know yet. So you'll have to just wait and see. My guess is that if we are able to, that would be fantastic. But I think our chances of being able to identify this are going to be fairly slim. Shell disease has turned into a much trickier disease to study than anyone I think really thought it would be. So I think identifying this causative agent might be pretty difficult. Again, we're going to be doing a lot more work with this. There's still a lot that our collaborators are going to be doing with all of these DNA samples. We've also done a couple of other things, sort of just because we had the opportunity to. While we were looking at these animals, we also collected some blood, and we took the blood and we put it on some bacteria plates. And we're like, okay, well, let's see if any bacteria grows. Let's see if the lobster has bacteria on them. Surprisingly, some of them did actually show bacteria growth, which is somewhat unusual for lobster blood. It tends to be pretty sterile. So we are going to be identifying what those bacteria are and find out if they're important in the disease state that lobster was at when we drew the blood. We'll also be doing some histology, looking at the health of some of the tissues like the liver, the gills, and the gut. And our collaborators will also be doing some scanning electron microscopy. And basically, there's a whole bunch of other questions that we're planning on asking. How does bacteria on the healthy lobsters differ from one species to the next? How does it differ from our laboratory versus their laboratory? There's like a zillion questions that we can ask with the samples that we've collected. I mentioned the scanning electron microscopy. I don't know a whole lot about this, but they sent me some really awesome photos, so I thought I'd share them with you. So this is a lobster shell with some disease on it. This is that same area of shell, and this is a scanning electron micrograph of it. So basically what you're seeing is a shell disease lesion right here. What's really interesting about this photo is you can also see these little pits over here. This is not disease, this is actually just the way the lobster shell is. So a lobster shell actually has a lot of pores and canals in it, which most people don't realize. So it's much more complicated than you kind of just look at it and you're like, oh, it's a shell. But there's a lot of topography to it. This same area, sort of magnified, this is the lesion, this is a healthy part of the shell. And they very nicely labeled this one for me because I had no idea what I was looking at. This is, again, healthy shell and some bacteria that you're seeing on the shell. So the whole purpose of all this, sort of to inform you, with shell disease is that it's a really complicated disease. And as I mentioned before, it's a multifactorial disease. There's all different kinds of things that need to come into play for a lobster to get shell disease. There's not going to be just one thing. You do need that one causative bacteria, but it's really going to be a suite of environmental factors, lobster health, and that pathogen in order to get this sort of disease level. So it's that whole Goldilocks theory. You really need just the right conditions, both of the lobster and the environment and that bacteria for us to start getting shell disease. We study shell disease not only because we think it's important, it's really interesting, but also because, you know, as humans affect our environment around us, we're also affecting those animals that live in that environment. And it's more important for us to find out, to learn not only about the diseases that affect these animals that we may be causing, but also about the animals themselves. Because even though this disease itself might not be causing a lot of mortality in the wild, the next disease might. And the more we understand about the animals and how we affect these animals, the better we will be prepared to learn about the next disease. There's a whole bunch of people I need to acknowledge with this work. Dr. Miranda Walker is the postdoc over at Swansea who has been the major collaborator on this project. Dr. Andrew Rowley is her principal investigator. Charlotte Davies, a master's student who I think is doing a lot of the DNA analysis that I'm talking about. My boss here, Dr. Michael Telesti, who also has been instrumental in getting all this work done. The funding agency over in the UK. And none of this work is possible without my group of volunteers and interns. These guys, one, keep me sane, two, keep me from having to take care of lobsters day in and day out, 7 days a week. And a lot of them have been doing a lot of this work. Some of the pictures that you saw were taken by them. They take a lot of the measurements, and they really keep the lab running for me. And that's it. I'll take any questions.